So looks like it's a laptop. Yeah. Should be.
Like really unresponsive.
Yeah. Live. So right now the broadcast is going.
Yep.
Just set it back to the power.
Yeah.
Not bad. There we go.
Yeah.
Wait, do you bring a laptop?
Why? Sure. Hello. Hi, everybody. Most of you don't know who I am, but some of you might recognize me as the girl who eats all of Dr. Zhu's food. I'm also running for Governing Council, U of T, and Governing Council is an association made up of students, faculty members, alumni, and staff who basically discuss certain propositions brought forth. So they handle a lot of the financial and services offered here at U of T. So I think it's of great importance to you that you do vote for this event. I forgot. Oh, as a student, employee, and oh my God, as a student, employee, and sister, I think I could offer the opportunity to represent a whole variety of the student populace here at U of T. So I'd love it if you could all vote for me. I'm Eglantina, and as Dr. Zhu likes to point out, the way to your hearts is through your stomach, so I brought some eggs to remind you all to vote for me for Governing Council. I'll put them at the front.
Okay, folks, if you don't mind. Just a really quick note about the midterm. There are a couple of quick notes, actually. The first is that this lecture is not on the midterm, nor is the tutorial material going to be tested on your midterm. The other thing that I wanted to point out to you, and I haven't made an announcement about this officially, but I will on the announcements page for everyone in case you missed this announcement or if you aren't in lecture. I went over the midterm very briefly with some colleagues of mine and some other individuals. And I had originally told you that your midterm was going to have 26 multiple choice test questions and 3 short answers. Is that correct? And I also said that it was going to be a fair midterm. 2 out of those 3 are going to stay the same. 1 of those is not going to stay the same. I'll keep it fair, We'll still have three short answers. One being like a diagram, another one being relatively short definitions, and another short answer. But in order to give you time and to make it a fair process for you to be able to check your answers before you submit your midterm, I found it a little bit long, so I decided to cut down the number of multiple choice test questions to 22. So you'll only have 22 22 multiple choice and 3 short answer questions, just so that you do have some time at the end to be able to check your final multiple choice test questions before you submit them. Next week not next week, apologies. The following week in tutorial, you'll actually have a short diagnostic midterm that you'll run in your tutorial. Then you'll have it taken up by your TA. It's not for marks, but it'll give you some indication about how prepared you are for the style of questions that you're going to get on Friday. Do you have any questions about the midterm before we go on? No? Okay. So the next sort of block of lectures that we're going to go through, and this sort of goes back to what we originally covered with the gut microbiome and other things, is related to choices. You choose whether or not you're going to eat red meat. You choose whether or not you're going to eat processed meat. And the next section is you choose whether or not you're going to be physically active or physically inactive. And both of those are lifestyle choices. You do not have to, by any definition, eat meat or red meat. You can eat poultry, fish, whatever else, if it's affordable for you. And you should be engaging in physical exercise. Both of those have some serious consequences on your biology. And this is sort of what our next topics are before we switch gears and then we start talking about things related to bioethics and research. But you're actually choosing for yourself whether or not you decide that you're going to give yourself cancer down the road. If you have the background and you have the genetic background, you're more likely to give yourself cancer, as some of you have been reading in your tutorial notes related to this Lancet Oncology article that came out in October of 2015. So I want to go through a couple of things with you today as an overview, and then we'll come back and revisit a paper very briefly on Friday, and then we'll wrap things up for the section when you come back after the break. But I'm going to go through a couple of things related to red meat as a carcinogen, and it is defined now as a carcinogen. And it goes back to this paper that was released in October of 2015. Again, most of you have been reading up a little bit about this in the Toronto Star article, and this is that paper that they were specifically mentioning. It's only 2 pages long, and it's kind of an interesting read to go through what the 22 different scientists actually talked about in that particular paper. But based on their findings, this made news because officially red meat now became classified by the World Health Organization as a Class 2A carcinogen, which meant that, as you read, it's likely a carcinogen. It does not mean that it is definitively a carcinogen, but it's likely a carcinogen if you have other things involved in your genetics, etc. We know that processed meats, so meats that actually have been cured using chemicals or have additional chemicals added to it, we know that processed meats were actually in that same paper, in that Lancet Oncology paper that I just showed you, are causes of cancer. They are put in the same class, and it doesn't mean that you are going to have just as many cases of cancer, but it's just the same type of chemical that causes lung cancer as asbestos causes, or air pollution, HIV, tobacco, x-rays, alcohol. And we know that in terms of our own biology, that overall over the last 40 years, that our meat consumption has gone up dramatically. And in particular, our red meat consumption overall as a population, I'm not talking about individuals in this class, but if we looked at how much meat everyone in this class ate compared to 40 years ago, the amount of red meat in our diets has increased quite dramatically. It's over half of our diet consists of red meat. So Interestingly, if we also look as a correlational study, it is not causational, it is a correlational study, the incidence of colorectal cancer has also over the last 40 years gone up. And I'm going to try to make the case that perhaps this is one of the reasons and one of the findings from the World Health Organization, looking at a number of different studies, including the types of studies up here, looking at observational studies, looking at the incidence of colorectal cancer, or CRC as I'll abbreviate it today, and red meat consumption in different populations versus doing things actually in experimental models versus looking at different genes that are associated with different types of cancers, that overall the findings from that 2015 paper showed that there was probably, based on observational studies that they looked at, and they looked at over 800 of them, that there was a strong likelihood that colorectal cancer was somehow associated with red meat consumption. And they can't really make much more than that type of statement because they haven't done as many animal studies as these observational studies that they looked at earlier on. But those are some of the reasons why we're looking at this in this particular context. So I wanted to mention a couple of things really briefly about colorectal cancer. And again, I'll abbreviate colorectal cancer sort of as CRC. It is not a trivial problem. If we look at cancer as a whole, we know that cancer is something that will impact every single one of us in this room. I'm not talking about colorectal cancer. Every single one of us in this room will either have a relative or yourself will directly be impacted by cancer sometime during your lifetime. And we know that when we look at colorectal cancer, which has been increasing steadily over the last 40 years, that currently in 2016, when we take a look at colorectal cancer, this is actually the third most common cancer that we will encounter here in Canada. In the United States and Canada and in many westernized countries, this is the third most common cancer in both men as well as women. It is a diet-related phenomenon, and so it's not unusual that we have it equally in both men as well as in women. If we take a look at the overall statistics of the Western world, and I'm going to show you some other statistics in the United States, but overall in the entire Western hemisphere, including Canada, the United States, parts of Europe, this is actually the second leading cause of cancer-related deaths. So this, again, is not a trivial sort of disease or disorder to take a look at. The interesting thing about all of this is that it is imminently preventable. So think about all those deaths, the second leading cause of cancer-related deaths, and it's something that can be prevented as long as you get screened for it earlier on. And I'll talk a little bit about screening in some more detail in a few slides. But this is the reason why we're interested in the biology. So yes, there's a correlation with meat, and red meat in particular. Yes, it is a very impactful disease, but it's also a preventable disease. If there's any kind of disease that you can prevent, it's actually something that we should be studying in a lot of detail. Again, you don't need to worry about the statistics that are on this slide, but if you were to take a look at the overall incidence of colorectal cancer, and it's just for your own information, in the United States and in Canada, and we're not talking about all of the Western world, the statistics are similar but a little bit different. But if we were to take a look at the overall statistics, we would see that colorectal cancer is right up there in terms of the number of new cases that get detected in the clinic. So colorectal cancer is right up there with things like prostate cancer, as well as with lung cancer and others. And we know that in terms of the related deaths due to colon cancer or colorectal cancer, that again, in this particular graph, looking just at the United States and Canada, that it is the number 3, and if we take the entire westernized world, the number 2 cause of cancer-related deaths. And so it's right up there. It's not something that you hear a lot about. It's not related to like cancer, etc. We talk a lot about breast cancer, and as we well should, but in terms of the overall impact of colorectal cancer, this is a very big disorder. And it's something that, in many instances, is caused by something that we're ingesting, and also because we're not taking proper care of ourselves. Your parents, by the way, who are likely in their late 40s and early 50s, are at the age right now where they should be routinely screened for colorectal cancer. So does it have a high incidence? Yes. Is it something that is preventable? Absolutely. Is it something that your parents are at all worried about? Probably not, to be honest. But should they be? Yes, 100%. So in terms of the overall physiology and anatomy, the colon, this structure that we're going to be looking at here, also referred to as the large bowel or the large intestine. This is, again, part of your digestive tract. It's fairly short. It's only about 5 feet long. Your small intestine is much longer than the large intestine or the colon. And its major function is really as a place where you can reabsorb some excess water that hasn't been absorbed by the small intestine, and it's, again, there to absorb some salts, some minerals, some vitamins as well. And what we've been looking at is this is where we find the richest diversity of bacteria, and our gut microbiota is here within this area of the colorectal tract. The last 6 inches or so of this is the rectum, and all of this entire structure that we're looking at, whether it's the transverse colon or the descending colon or the rectum, all of this, in theory, can have cancer associated with it. So 5 feet doesn't sound like it's a lot, but being able to diagnose and to be able to examine that entire 5 feet is something that, in terms of medical science, right now we can do that. And we can actually make sure that we can monitor this on a routine basis, but the vast majority of individuals are not going to go. And I'll show you the reasons why your parents are not likely to go until it's almost too late. So what are some of the risk factors? Age. Age is the #1 risk factor. As you get older, every single one of you sitting in this room has an increasing probability and an increasing likelihood that you're going to develop colorectal cancer. And your parents are now typically in that age range of being over 50 years of age, where they're at the highest risk of having colorectal cancer. Some of you may know, if you have a history of colorectal cancer in your family, they might have started off being polyps, small growths, or adenomas, and I'll show you that in a few moments. If you have other forms of inflammatory bowel disease in your family, for example, if you have had in your family forms of ulcerative colitis, where the colon itself becomes ulcerated and parts of it are being degenerated, and it's also undergoing inflammation, or you have Crohn's disease, which is very similar, then you are also at an increased risk of developing colorectal cancer. One of the things, again, to just differentiate this, some of you will have heard of irritable bowel syndrome, or IBS. And IBS or IBD is not associated in any way with colorectal cancer. However, some of these other inflammatory bowel diseases certainly are. So they put you at increased risk. If you have someone in your family that has this, or you have had this, or you've been diagnosed with one of these things, then you're at increased risk of developing colorectal cancer. But all of us are going to age. Every last one of us is going to age, and we're all going to be at that point where we have an increased risk of developing colorectal cancer. Certain races and ethnicities also have an increased risk of developing colorectal cancer. There is definitely a genetic component, which we'll come back to on Friday and talk about APC as one of those genes that are involved in colorectal cancer. We know that there are some other inherited genetic disorders that also increase the likelihood that you are going to have colorectal cancer. And these are, by the way, I label these sort of as intrinsic. There's nothing you can do about this. You can't change your genetics. You can't change your age. These are intrinsic factors that are beyond your control. These are things that you're born with, or that you are going to accumulate over time. And so as intrinsic factors, we can't manipulate these whatsoever. So again, other types of things, your genetics, if you have other inherited syndromes like FAP or HMB204 and you've been diagnosed with these, then you're also at increased risk of colorectal cancer as well. And these are all intrinsic factors, whether it's related to your DNA that you've inherited, your age or your aging process, your race or ethnicity. Those are all intrinsic factors that are beyond your control. However, there's also this component, which is the extrinsic component to colorectal cancer as a risk. And your diet is #1 on there. So if you know in your diet that you, sorry, let me just try to get the laser pointer back, and apologies on this. We know that if in your diet that you are taking in a lot of processed meats, and we're talking about processed meats like pepperoni, salami, all those really great tasting meats. If you're doing and increasing your daily intake, weekly intake of those, that you're putting yourself at an extrinsic risk above and beyond what you would be able to encounter only by your genetics, only by your ethnicity, etc. And this is something that we can control. We can't control our intrinsic factors, but we can control our extrinsic ones. So red meat, and having red meat in our diet, that all of us have seen as increasing in Western society, as well as processed meats, all increase the risk of developing colorectal cancer. The way in which you prepare your food also puts you at increased risk. So it's not only that you have red meat, but if you are cooking these at really high temperatures or barbecuing your meat, that also puts you at an increased risk as well. If you are not eating as much in terms of vegetables and fruit, this is also putting you at increased risk as well. So if you decrease the amount of fruits and vegetables that you're intaking as a choice, or maybe it's beyond your choice, it's getting too expensive to do that, that's also an extrinsic risk factor. Physical inactivity, right? So when is the last time you hit the gym? When's the last time you went out and played some basketball? When's the last time you were physically active? That is a choice. That is an extrinsic factor. And I can tell you that all of you are busy, right? And all of you are really busy with studying and everything else. But it's a choice whether or not you spend 30 minutes going to the gym every other day, or deciding that it's better to not do that. And that, again, is an extrinsic choice that we are deciding as a increased risk factor for developing colorectal cancer. We've talked about other things. Is obesity related to diet? Absolutely. But obesity itself also increases the risk of developing colorectal cancer. And you can see how number 1 and number 2 is tied into obesity. Smoking and alcohol consumption also dramatically increases the risk of you developing colorectal cancer. Those are choices. Those are ones that you can decide as an extrinsic factor to simply avoid doing. Type 2 diabetes related to metabolic syndrome, also related to both factor 1 diet as well as physical inactivity. So you can see how all of the different themes that we've looked at in the course are kind of tied together. That overall, these are lifestyle choices that we're making that directly impact our biology, and as a result of impacting our biology, is impacting our health status and disease status. So again, I had sort of talked about this in the last lecture, and I'm not going to spend a lot of time on this. But usually when we talk about red meat or processed meat being risk factors, some of it is really the byproducts of the meat. We know, for example, that if you are processing the meat, either through salting, curing, fermenting, smoking, etc., to either enhance the flavor or to make the meat last longer, that those are going to produce different types of chemicals that I'll show you in a few moments that have been shown in animal models and in observational studies to be deleterious and an increased risk factor for colorectal cancer. In particular, things like N-nitroso compounds, or N-nitroso just as an abbreviation, as well as the generation of these polycyclic aromatic hydrocarbons, depending on the type of cooking choice that we engage in, will increase the risk of colorectal cancer. And cooking certainly makes meat taste better, increases the digestibility. It's easier for us to chew it as well as to digest it, but it also produces a lot of chemicals in addition to these and nitroso compounds and polycyclic aromatic hydrocarbons. It will also produce these heterocyclic aromatic amines as well. And so we know that cooking by itself and the way in which we cook things will also produce some deleterious chemicals. And again, there are a number of ways that we cook our meat, whether it's pan frying, grilling, barbecuing, etc. These will increase the likelihood of producing these chemicals, and as a result, increasing the likelihood or the risk factors associated with red meat. And again, red meat by itself is not necessarily a bad thing. We do get vitamins like B vitamins, as well as having a good source of iron, as well as zinc, and we get lots of our protein in our diet from meat. And if you've ever wondered why are there so many protein bars that are out there, I think one of the reasons is simply for convenience. If you've gone to work out and you need to replenish protein, protein bars are a great source for that. But another reason is for precisely these reasons, that we don't need to ingest protein only through meat, and having these protein alternatives has become an attractive source as well. So if, for example, you've had these risk factors, you have intrinsic risk factors and you also have extrinsic risk factors. One of the things that we know happens that in some individuals, not all individuals, but in some individuals, the changes in the lifestyle as well as in the choice of food will develop an abnormal growth, which we call a polyp or an adenoma. And I'll show you the adenoma on the next slide. And a polyp is this growth of a tissue in the lining of an organ, and in this case, the lining is part of the colorectal tract, okay, or the colon in this particular case. And there are two types of these colorectal polyps. One is hyperplastic, and the other one is ones that will form an adenoma. And the hyperplastic ones, even though the name sounds like hyperplasia, these are ones that are unlikely to develop into a full-scale tumor. The ones that form an adenoma, however, are the ones that will eventually, as they continue to grow, so you start off with a small growth, a polyp, the polyp grows bigger, forms an adenoma, and then this adenoma will then form a tumor. These are all things that you can actually get screened for early. If you eliminate this polyp at a very early stage, you can actually prevent that cancer, which is why we say that this is a preventable form of cancer. So your normal colon, the lining of your normal colon, as you put in the colonoscope, will actually show the smooth lining that you would normally expect. And you might start to see things like this adenoma. And it's very visible using colonoscopy to be able to observe this. And if this is allowed to continue to grow, this will eventually form this large mass, which is easily detectable as a form of colon cancer. It is a lot easier for you to have this excised and eliminated than it is to remove a section of your colon in terms of this form of colorectal cancer. So like all diseases, whether it's metabolic syndrome or if it's things like colorectal cancer, the earlier you can detect this, the better it is for you. And it's like a form of early prevention. And you can simply have this done through regular screening. Now, you'll hear a lot about this in the news, about regular screening, regular visits to get a colonoscopy done. And the reasons for this are on the next slide. And this screening is actually fairly easy to do. It's easy to actually detect this adenoma or this polyp at an early stage. And if you get rid of it at an early stage, you're unlikely to have it developing into a colorectal cancer. And the whole idea is with regular screening, because these polyps take time to grow. If you catch them early before they start spreading, they will not grow into a full-fledged cancer. And these different types of screens that I'm going to spend a few moments on in the next few slides are really important, this early form of prevention. Now again, take a look at this bottom slide. Why is Health Canada, why is the Ontario government so interested in getting you in when you're at about 50 years of age in for this early screening? Because these polyps that I've just shown you start off really, really small. And for them to develop, in most cases, there are some really aggressive forms of cancer. If, for example, you have a genetic background where you already have things like HMB204 in your family history, then you are going to have this timeline shortened. It's not going to take 10 years for it to develop from a polyp all the way into colorectal cancer. It may be half the time or a third of the time. But when you start early, and you see these as small adenomas or small precancerous polyps, and you remove them, then they're unlikely to develop into these larger forms of cancer. So these things take time. And as long as we're catching them at an early enough stage, then we can prevent this cancer from occurring. And there are a number of different tests and a number of different screens that allow us to get this early type of detection. And I'm going to go through them fairly quickly with you. And I want to make sure that, again, it's not for your midterm, which does not include this lecture, but for your own interest, as well as for the final exam, that you're comfortable with all of these different methods that I'm going to be covering for you. So I'm going to tell you the difference between looking just at the sigmoid portion of your colon, as well as a full colonoscopy. Some other techniques like double contrast barium enemas, as well as CT colonography. Now, all of these are designed to look for abnormal growths within your colon. And some are a little bit better, some are a little bit more sensitive. We won't go through all of the fine details. But again, the overall idea is if we detect these early, we can remove these polyps or these adenomas before they become full-grown cancers. And the screening allows for that individual to not have cancer later on in their lives. So again, this slide actually shows you both a sigmoidoscopy here at the top, as well as a full-blown colonoscopy. They're really the same thing. The sigmoidoscopy is really just examining the lower third or so of your colon, and a colonoscopy examines the entire length, all 5 feet of your colon that I showed you earlier on. And this sigmoidoscopy is often used, for example, if you have a family history, especially in the rectal area, or if you have other forms of irritable bowel syndrome, etc., to use. But overall, the idea is exactly the same for both the sigmoid as well as the full-blown colonoscopy. You take in and put in a flexible lighted tube into the rectal and anal canal up through the entire length of the colon, and you're just simply looking for abnormal growths. And again, they're fairly easy to see. And then built in to this sort of probe, not only does it have a light, but it also has a way to remove these different polyps that I'll show you on the next slide. But the reason why we can remove them is not simply to get rid of the growth, but also to remove them so that they can be further processed for histopathology as a form of biopsy. So this is the probe that goes in. If you detect a growth, so a light is also shining, and if you happen to detect a growth, you can actually put out a metallic platinum wire lasso, and you can actually cut off the growing polyp that you've identified. And again, as a form of early detection, this is really useful. This tissue that gets removed or excised is then captured by the probe, removed from your body, put into, they'll look at the sections of the polyp, and then they'll be able to further diagnose whether it has become cancerous or is still at the precancerous stage. So this is one of the methods that we use for screening. Another one is this barium enema, and barium is a substance that is X-ray opaque. X-rays can't really pass through it well. If you inject this into the colon, it allows you to be able to see the structure of the colon really well, and this white is where you have put in the barium contrast medium. And it's the same idea. You introduce this barium contrast medium in through the rectal region, and after that you can then put in X-rays. The X-rays will then allow you to see if there are any abnormal growths or polyps that are protruding in to the white region that you can see that lines the inside of the colon. And all of these are effective. All of them are a little bit different, but all of these are really effective screens to see if there's any sort of abnormal growth. All of these are designed to see either a polyp or colon cancer within this tissue. So the last one is this CT colonoscopy. And here it's a slightly different. Same idea. We want to be able to visualize any abnormal growths within the colon, but here we're pumping in air, and as air gets pumped in, one of the things that happens is it allows the colon to expand, and we can then take thin X-rays. And again, in X-rays, air is something that is not dense. It doesn't appear white. It actually appears black. And so as a result of this, because we've now blown up your colon with as much air as possible, we can take a look and see, using these X-rays, to see if there's anything protruding into that lumen that you see here. So this is the lumen of the colon where air has been put in. Air also, again, reacts with X-rays in a certain way. And we can actually, because it's a thin CT scan, develop a 3D model of the entire colon, and we can actually start to see if there are any polyps or colorectal cancer as well. And these are two different types of CT scan models that have been built up around this. So those are different visualization techniques or screens that people can use to look for colon cancer or colorectal cancer. And then there are some biochemical tests that can also be used as well. So one of these is known as the Guillac fecal occult blood test, and occult here meaning hidden, right? So your fecal matter, your stool sample, your crap, whatever you want to call it, actually has, if it has blood in it and it's hidden, you can't see it, that is not a good sign. And there are lots of reasons. Your parents, for example, might be complaining that they have a lot of pain in their abdomen, or after they've eaten, they feel a lot of pain. They might be feeling like they are less energetic. They might have anemia for whatever reasons, and they're bleeding into their gut. They might be bleeding into their gut because they have something growing there. And this will tell you if you are bleeding into your gut. And this involves a particular type of paper, Guillac paper, and this is a phenolic compound, and it's named after the particular acid. And when you have your stool sample on this phenolic paper, and you add acid to it, if it turns blue, that means that you have blood in your stool. It's a very quick way to see if there is any blood at all in your stool. These other two tests actually tell you this same thing in a slightly different way. And again, we won't go into a lot of details about this Guillac fecal occult blood test, but again, you take a stool sample, or your doctor takes a stool sample, and it can be due to a number of things, right? So suddenly your parents, even though they might have been eating well and eating exactly the same as they normally have, start to lose weight. Or if they suddenly have abdominal pain that's just coming out of nowhere, they can't come up with a good reason why they start having this abdominal pain. If there are changes in the bowel movements, so normally they're fairly regular, now they're no longer regular, or there is a difference in the type of bowel evacuations they have, all of these things might be due to colon cancer. It's not the only things that cause it, but these are things that might cause it. Anemia, low blood count. So if you are suddenly not feeling as energetic as you are before because you have anemia, and it's not due to your diet, but it's due to you bleeding out somewhere that you can't detect, this is another reason why this might occur. In some instances, if you have been bleeding into your gut for a very, very long time, your stool itself, the evacuated fecal matter, is going to either look really black or really tarry, and it's not the same appearance that it normally has. That's another reason to get this test done. And then again, we take 3 different bowel movements just to make sure that it is something that is consistent. And again, we run through this test, and you can see really quickly when you have a blue color that you have blood in your stool. And then it's up to you what you decide to do next, and we'll talk about that at the very end. So in addition to this, we can also look for immunochemical tests. We don't have to necessarily just use that Guillac paper test. We can also use an immunochemical test where we have antibodies against hemoglobin. Hemoglobin is part of your blood. Normally we don't have that in our stool, but now in your stool, if you have hemoglobin in there, that likely means that you're bleeding out into your colon. And so we have this FIT immunochemical test for hemoglobin as well. And let me ask you this. What would you do a DNA test on in your stool? What are you going to be looking for in terms of DNA? Yes. Sorry, might be some cancer cells. So how would you test for that? You're right. So might be cancer cells are being, as part of a colorectal cancer, are now going to be detected here. What would you look for? Too many chromosomes? Possibly. Any other ideas? So we are looking for cancer as part of this DNA test. It's not like it's not a normal part of our body. Yes. You can do karyotypes, as your colleague mentioned at the front, but we have a panel of identified genetic abnormalities that you would be tested for. And if we ran that panel using this DNA test on your stool, and we found that there were markers for abnormal genes or chromosomes that were associated with colorectal cancer, then you would be sent for secondary screening. And again, one of the reasons we like doing this is it tells us right away what types of cancer might be evident. And again, it looks a lot like this FIT test, for example, is really easy, quick to carry out. You can actually do this at home if you are so inclined. It's a little messy, so I probably suggest you don't do it at home if you don't have to. But it looks a lot like sort of an ovulation or a pregnancy test. You get like 2 lines or 1 line, and it's very clear like if you have hemoglobin in your blood. So there are other ways to go back and test for this as well. All of these colonoscopy findings, though, are actually really important, because if any of those things came back positive, right? So if my FIT results came back positive, or my fecal occult blood test came back positive, there are a number of things that can actually cause that. I would still have to go in for a colonoscopy, and I would still have to use a colonoscopy as the definitive whether or not I have CRC test or not. And any type of abnormal finding has to be followed up using colonoscopy. So when should you get this done? So at the age of 50, so unless you have other predisposing conditions. So at the age of 50, both men and women, so hopefully now you understand that it affects men and women equally. And we know that it's the 3rd leading sort of type of cancer, as well as the 2nd leading cause of death related to cancer, that both men and women should start to get screened. It's not just men that should be screened for colorectal cancer. It's both men as well as women. And again, this is a great test because it looks for both polyps as well as the presence of cancer. These things take time to grow. So if it came out negative, you had it done at the age of 50, it came out negative, you don't have to have one the next year. You could wait a few years before having this done again. You don't need it every single year. And a colonoscopy can be done if it's a negative finding over 10 years, as long as you have no genetic background for this. That flexible sigmoidoscopy, for example, you might want to do it fairly more routinely every 5 years. But overall, these are fairly innocuous tests that are easy to do. It's only a local anesthetic that you're given. You go in, you are ambulatory, you can walk out after a few hours as well. If you have an average risk of colorectal cancer, what does that mean to you, by the way, an average risk? It's not an A+ risk. It's not a B+ risk. It's just a C risk. What is that? What is an average risk? So let me ask you this question then. So my father had colorectal cancer. He actually had colorectal cancer that infiltrated past his colon, and it was found in some of his lymph nodes, and he had that when he was fairly old. So am I at increased risk? So am I average risk, or am I at increased risk? Increased risk. And so there's probably some genetic component to this. And for example, you're at an average risk if, for example, you have maybe experienced some health issues or anything else, but the genetic background isn't something that you're too concerned with. And again, here, you're getting screened on a fairly routine basis as well, but in slightly different ways. Because these are relatively fast and quick and can be done in your doctor's office. You don't have to go to the hospital. You can't get the colonoscopy done. You can't get a barium enema. You don't have x-rays typically at your family doctor's office. But all of these tests that you see here can be done within your doctor's office and sometimes even in your own home, so that these are really good to detect if you have things like cancer really quickly. But these are sort of the things that I want you to be thinking about throughout sort of Friday as well as in your own lives, right? So these are serious disorders. These are disorders that have intrinsic factors, which are interesting to us as human biologists, but they also have extrinsic factors. You are choosing whether or not you are going to have a disorder down the road. What you eat now, what you decide to do or not to do, engage in physical activity or not, which is the next module that we'll talk about. All of those are choices. And you have a choice every single day to either eat healthy or not eat healthy, to engage in exercise or not, to go for screening. That's also your choice. No one is forcing you to go. No one's forcing your parents. I'm sure they're just like me. They're really stubborn. They don't want to go see the doctor, and they're like, it's okay. It'll go away with time. It's up to you to convince your parents that maybe that little twinge that they feel is something that they should get checked up on at an earlier age. So we're going to end a little bit early today. For those of you who are writing your quiz, good luck on that. And next week, no class, but we will have a tutorial session on Tuesday.